Today we're going to take a look at the different structures of proteins and a better way of understanding them. Now there are four different structures: the primary, the secondary, the tertiary, and the quaternary. The primary is going to be just a string of amino acids. So I've got a string right here. So this is going to be all the different amino acids from alanine to phenylalanine. There's not much of a structure to it except for the dipeptide bonds between the amino acids. This is the primary structure, or the one-dimensional structure. If you've ever taken an art course, you would know that one dimension typically just means a line. Now, the second dimension is going to refer to the various shapes we can make. For proteins, we have two different structures. There is the alpha helix, which kind of looks like a spring. And then we have the beta pleated sheets. Now, obviously, I'm not the best artist, but those are the general secondary structures for proteins. But how can we apply that to our line over here? What we're going to do is I'm going to make another line over here. And we're going to just add a few extra lines. So it's not the prettiest picture, but we see that it's taking form. Alright. So now I have a bottle of water. This is our secondary structure, our 2D structure. This is where the proteins make their first shapes. So that gets us through our first two shapes. But we need to add another dimension to it, our tertiary structure, also known as our 3D structure. And here we have our tertiary structure. This is where we've made a three-dimensional image of the object. So this is where you can actually see what it looks like instead of just some general shape. This is going to be when we fold all those alpha helices and all those beta pleated sheets on top of each other to form a three-dimensional structure. Now you may be asking yourselves, wait, that's a 3D shape, and you said there were four structures. How can we have a fourth dimension added to this? Are we going to break the space-time continuum? Unfortunately, no, we're not going to do that. What the quaternary structure is going to do is it's going to take this one object and add several of the objects together. So for a water bottle, think about having a 24-pack of these water bottles. Each individual water bottle is the tertiary structure of the protein. Now we have taken one protein and added a bunch of other proteins together to make a much bigger protein that can have multiple functions. In fact, a lot of the proteins that we're going to study in our course have quaternary structures. For example, hemoglobin is going to have a couple different subunits of proteins. I hope this helps your understanding on the protein structures.